Hey guys, it's Stratton. And today's video is on evidence of evolution, which is AP topics 7.6 through 7.8 and 7.11 through 7.13. So what we're going to talk about today is how do we know that evolution happened and how do we know that it continues to happen today? First, I want to go over the RP from last time, which was on Hardy-Weinberg equilibrium. So the first problem, frequency of the curly-haired allele, big H, is 60%. So what's the frequency of the straight-haired allele, little h? Well, if this is 0.6, then that means that Q is going to be 0.4. Calculate the frequency of the population, or percentage of the population that has straight hair. So straight hair is the recessive condition, so this is going to be Q squared. So that's going to be 0.4 squared, 0.16. And then calculate the percentage of the population that has wavy hair. So this is an incompletely dominant gene. So this is going to be 2pq, which is 2 times 0.6 times 0.4, which is going to be, let's see, 0.48, so 48%. Okay, number 2. 9% of the population of Tristan da Cunha is afflicted with the recessive disease retinitis pigmentosa. Causes blindness. So the frequency of the dominant recessive alleles. So what they gave me here, they don't have the word allele in there anywhere in the prompt. So 9%, so 0.09 is going to be Q squared. So it looks like 0.0Q, but so this is Q squared is 0.09. So the frequency of the recessive allele then would be Q is the square root of 0.09, so that's going to be 0.3. That means P is going to be the rest of it, which is 0.70. And carriers would be, of course, 2PQ, which is going to be 0.42. So that was 2 times 0.3 times 0.7. Okay, within the population of butterflies, brown is dominant over white. 13% of the butterflies are white. So that right there, they gave me this 13% is my Q squared is 0.13. So that means then that Q is going to be equal to the square root of 0.13, which is 0.36. So P obviously is going to be equal to 0.64. So a percentage of butterflies that are heterozygous, 2 times 0.64 times 0.36. And that's going to be equal to 0.46. And then the homozygous dominant genotype, that would be P squared is 0.64 squared, which is 0.41. Okay, hopefully you got those. All right, I went ahead and took the liberty of filling in this table here. And what we're really looking at, of course, is the change from 1985 to 2011. So the change from 0.48 up here to 0.78, that's a change, an increase of 0.3, and then of course those alleles here, this decreased by 0.3 because they both add up to 1 obviously. So what it's telling me is that the big E allele, which causes early migration, increased in that time period. So what were the scientists testing? What hypothesis? It was likely what is the effect of changing temperatures on the allele frequencies. If you look, you've got the mean annual water temperature in degrees Celsius there, and a change from 6.2 degrees Celsius to 8.4 degrees Celsius. That's more than 2 degrees Celsius, which is 5 degrees Fahrenheit or so, and that's a pretty significant change. It caused earlier migration. So based on the graph, propose an explanation. So in my estimation, I would say that increased water temperature is selecting for earlier migration. The fish are able to get into those streams a little bit earlier and lay their eggs, and the offspring are able to start growing earlier in the season. They have an advantage, higher fitness, and the justification there is that the data supports this. There's a correlation of 0.7866, which says that water temperature and the frequency of the allele are likely related. Okay. So today we're going to be talking about types of evidence for evolution. The first of those is geographical. Then we'll go into geological, physical, biochemical, and direct observation of evolution. Despite what some people say, we actually have seen evolution take place in the lab and in the real world. So we'll start with geographical evidence. Number one, we know today the continents are not in the positions they've always been in, and they're constantly moving, and earthquakes are a symptom of that. They didn't always know that. In fact, it wasn't until the mid-20th century that they finally accepted the evidence that there were these plate boundaries that were moving, and that the plates were actually floating around on this sea of magma down inside the mantle of the Earth. So the continents are not where they've always been, and the result is that the climates have changed. In fact, Antarctica used to be tropical, and there are fossilized palm trees underneath, in some cases, a mile of ice. And so the study of the locations of the different fossils and organisms and stuff is called biogeography. And what we find is that the continents that were more recently connected share more similar species and have more similar fossils as well. So you can see there that obviously South America and Africa fit together nicely like a puzzle because they were connected. Antarctica, Australia, and interestingly enough, India and Madagascar were also down there, part of that lower continent, which I'll get to in just a second here. But they do share more similar species than, say, North America. So 240 million years ago, this is what the Earth looked like, approximately. So Africa and South America are located down here at the bottom. So the border is right along there. Australia and Antarctica down here, India, and the top, North America, California, we're up here pretty much underwater at that point. If you fast forward a little bit, 200 million years ago, you can see that you can start seeing the shapes of the continents there as they start to break apart. The upper and lower continents were pretty much separated at this point, one little connection between them, and those are the names they've given them, Laurasia and Gondwanaland. And of course, there is a movement right now to reunite Gondwanaland. Okay, 150 million years ago, you start to see more familiar outlines now. North America is starting to take shape, and this is about the time when dinosaurs were really taking over. They really were getting their start. And the dinosaur fossils you find in South America are not the same as the dinosaur fossils you find up in North America and Asia. Okay, the other thing that happened, to get a little bit further out here, is the Earth got a lot warmer. And as the Earth got a lot warmer, sea levels rose. And you can see the entire central part of North America up here is just one big sea. And the result is that all this area here today is made of sandstone. It's because sandstone is from where the ocean bottom used to be, and you find a lot of marine fossils there. And what's really important for our economy, and unfortunately pretty terrible for the Earth, is it's also where you find oil, because oil is primarily phytoplankton that were fossilized and then embedded underneath these sandstone rocks. We drill down today to get those out and then burn them and put ancient carbon back into the air. So when was it in the air? When the Earth was hot. What's happening is we put it back into the air, the Earth's getting hotter again. So dinosaurs were peaking at this time. And if we go forward to 65 million years ago, of course, that's when there was the impact. The Chicxulub crater is located right about here, the Yucatan Peninsula, and dinosaurs went extinct at this point. Now, mammals were starting to rise before this point. So there are mammal fossils all over the place. And in fact, if I go back to here, mammals were actually evolving about this point. And interestingly enough, you find that there's a different set of mammals that evolved up here than the Gondwanaland mammals. The Gondwanaland mammals, you're looking at things like marsupials and, well, originally, of course, egg-laying mammals. There's only 2 of those left on Earth, and those both exist in a former Gondwanaland country, which is Australia. But marsupials, there's only one native to North America, and that's the opossum, the Virginia opossum, whereas South America has some, and Australia, of course, has a ton. So there were very few placental mammals in Australia, really just ones that had managed to float there later, much later, or were brought by humans. So 65 million years ago, and then the Earth, all told, is 4.6 billion years old, but the first fossils that we have are about 3.5 billion years, And we're looking at things like stromatolites. That's what these are here. So the stromatolites, they were some of the first photosynthetic organisms, and they helped to convert our atmosphere and really changed the Earth. So the original oxygen that was created went into rusting the oceans, which is all where the red rocks that are all over the Earth come from. And then future oxygen collected in the atmosphere, and oxygen levels soared. So, and yes, sharks are older than trees. There are fossils that are 450 million years old for sharks, but trees are only about 350 million years old. So. Okay, number 2, geological evidence. So mainly we're talking fossils here. And we know from fossil evidence that there have been at least 5 mass extinctions during the last 500 million years. And out of the species that have existed on Earth, only about 0.1% are extant today. The other 99.9% are all extinct. And the interesting thing is that each one of these extinction events becomes an opportunity. It's a chance for organisms that did survive to branch out and go through what's called adaptive radiation and fill in the niches that become available as other things have gone extinct. So fossils, unfortunately, you're probably not going to get fossilized unless somehow you're buried in a landslide and nobody digs you out. Then who knows, maybe you will get fossilized. But fossilization is very rare. Very few organisms actually get fossilized. And the way that we figure out how old a fossil is based off of radioactive dating. So radioactive dating is when you look at how much of an isotope of a certain element should exist within a rock. And over time, as it breaks down, you can either discover that it's gone, or that it turns into a different isotope. So in the case of uranium turns into lead, and it takes 4.5 billion years for that to happen. That's how we know how old the Earth is, based off of some uranium-lead dating that's taken place. For more recent things, we can use carbon dating, but carbon-14 only has a half-life of a little shy of 6,000 years, about 5,700 years. And so you really can only date more recent things. If you were to die today and 10,000 years from now somebody dug you up, they would find that you had gone through about 2 half-lives. So you'd lost half of your carbon-14, and then about another half of your carbon-14. You'd have about a quarter of your carbon-14 left, and they would know, oh, this fossil is about 10,000 years old. There are lots of other elements that go through the same process, and so from that you can use a variety of different clocks, and you can confirm dates using several different isotopes, and it's very accurate, despite what some people would say. So Archaeopteryx was the picture that was on the first page there, and that is a fossil that was found in Germany. It's about 150 million years old, and it's a transitional fossil between dinosaurs and birds. Although, in fact, we know now that most dinosaurs probably had feathers, including T-Rex. And in fact, they've actually found some dinosaur feathers that were preserved in amber. So scales and feathers are very, very similar, and it's just one gene, one little mutation. You think about it, what does a chicken have on its feet? Scales. But there are mutant chickens out there that have feathered feet. Look it up. They're pretty interesting looking. Okay, when we're looking at whales, now whales are mammals, and they didn't really evolve until about 50 million years ago. The earliest ones, Opacacetus, named because it was found in Pakistan, and its nostrils were at the front of its face. It had full hind legs and everything. They've got fossils of this. And then today, the gray whale, which of course has its nostrils on the top of its head, we call it a blowhole, but it's really just its nostrils. So you would expect that somewhere in between you would have a transitional fossil. And that transitional fossil is Aetioceus, which has nostrils that are kind of halfway between the front and the top of the head. And it also has much smaller legs. By the way, you find plenty of these fossils in the middle of the Sahara Desert, because if you want to go back to one of those slides that I showed you earlier, it was covered by ocean. So here's the family tree. This is called a phylogenetic tree of whales. And what you'll see is that at the top you've got a very obviously land-dwelling mammal that probably could go into water for short stretches. And at the bottom you've got the 2 extant types of whales. So these 2 guys down here at the bottom. So the top one, these are your baleen whales. They're the filter feeders. And of course, you've got your toothed whales, like the killer whale at the bottom there. Those are both extant. Everything else on this chart is extinct. I will point out, though, if you were to come up here to the top, I could potentially add another branch off of here and come out to another extant animal that still exists. The closest relative, oops, sorry, let me go back there. The closest relative of whales, any guesses? It's the hippo. So this guy kind of looks like a hippo too, doesn't he? So the hippo is, based off DNA, the closest relative of whales. These are all the fossils they found. Well, not all the fossils, but these are some of the fossils they found of our ancestors. So you can see N is a modern human, and going backwards from there, you can see that the skulls get smaller because the brain cases are smaller, and the faces get a little bit more slanted, and these go back about 6 million years. Okay, physical evidence. These are structures that we have currently. So homologous structures are things that come from a common ancestor. In this case, the legs of the bones of the arms or legs or wings or whatever, pretty much every limbed animal out there share similar bone structure. The top bone being the humerus, the white one is the radius, the red one is the ulna, the carpals, metacarpals, and phalanges down at the bottom. And depending on which organism you're talking about, all it is a matter of how much those bones grow during development. And in fact, if you look at a horse today, horses are actually running on just one finger of their hand, the middle finger actually. And occasionally horses are born with extra bones sticking out the bottom. They usually remove those because they get in the way, but all the other bones are there. They've just been elongated and changed based off of development. Now, a non-example of this would be an analogous structure, something that doesn't come from the same ancestor but shares a common function. So for example, a bird wing, an insect wing, and a flying fish wing are all very different structures, and the only one that actually has these bones would be the bird wing. So next time you're eating a chicken wing, look at it and see how many bones are in that wing. It's possible you have the humerus, which is the one that has one bone in it, or it's also possible you might have the middle section of the arm that has a radius and the ulna has 2 bones in it. The hand is usually thrown away. So I took this picture at the Monterey Bay Aquarium. This is actually a gray whale skeleton, and I want to point out 2 features of this. Number one, it has the bones that I was just talking about, homologous to the bones of your arm. So the radius, the ulna, and of course the humerus at the top there. But it's also got something in the back called a vestigial structure. That vestigial structure is these 2 little bones that they've got suspended right here. Those are the remnants of a pelvis. Because whales, like I showed you earlier with the family tree there, with the phylogenetic tree, whales used to walk on land. And so vestigial structures are from the same ancestor or origin, but they're not used for that function anymore. So these obviously are not used for walking because this whale cannot walk on land. So be things like your appendix, which in other organisms, in particular organisms that are herbivorous, they use that area for the breakdown of cellulose and other materials like that. They're full of bacteria. Whereas ours, I won't say it doesn't have a function because it is useful for holding on to bacteria when you've got diarrhea and things like that, but it no longer serves a purpose for like the actual breakdown of cellulose. We really don't break down cellulose. It passes through us. We just call it dietary fiber. But there's lots of structures, and including down here at the bottom, I've got a video. I'm going to go ahead and put this up on Classroom as well. Proof of evolution you can find in your body. It's really interesting. It's pretty short. It's worth watching. But it can also include behaviors, and they do talk about that in the video as well, so it's worth checking out. Now, you always wonder what that little pink thing is in the corner of your eye? It's called the semilunar fold, but it's really a vestigial structure called a nictitating membrane. So nictitating membranes are used to protect the eye, and lots of organisms have them, including your dog. Next time your dog is sleeping, take a look, and maybe you'll see that its eye is partly open, but there's a membrane covering its eye. We just don't have that structure anymore. Just a vestigial version of it. Okay, also, your tail. Well, most of us aren't born with tails. I mean, we have a tailbone, and if you look at a human skeleton, the tailbone is very obvious, several inches long, but at one point it was fully 1/3 of your body length. So what happened? The gene for the development of that got turned off, and it just stopped developing. Otherwise, you'd have about a 2-foot tail. Okay, atavisms are structures that occasionally will get turned back on. That would be like a human tail. If you were born with a tail, that'd be an atavism. But it's things like every once in a while, a snake is born with legs. The genes are still there. Or a dolphin or a whale born with legs or back fins. For us, this is a little creepy, but we actually have what are called milk lines. Now, all animals have these. If you think about your dog has a line of nipples running down each side. Well, humans can too. It's just that those are turned off, and every once in a while they get turned back on. Now, it might just be a little spot, or it might be a full-on developed nipple. In this case, this guy has, 6 nipples? That's, yeah, a little disturbing. And then endosymbiotic theory, something we talked about earlier in the year, but the idea that bacteria were picked up by bigger bacteria and became the mitochondria and the chloroplast, and just more evidence for evolution. So, okay, number 4 is biochemical evidence, DNA. So yeah, you share 99.9% of your DNA with other humans, 98.7% with chimps, 90% with a cat, and even 60% with a banana. Now, I'm not saying that it's all these different genes. There's a lot of different DNA inside of you, but I mean, think about basic processes like breaking down glucose, right? Some of those enzymes, they're universal. So proteins, looking across here, in this case, like cytochrome C is the one that's most commonly used. So cytochrome C is basically identical between us and a chimpanzee. Thus in a gorilla, it's a little bit 98% similar, that kind of thing. And as you get further down the list, things become less and less common. Okay, metabolic pathways. So we share common metabolic pathways. In fact, this would be showing all these different possibilities for breaking down sugar, making sugar, fatty acids, and all kinds of stuff like that. So, and then going back to the Miller-Urey experiment that showed that the basic metabolism, basic metabolic chemicals of life can be made within that chamber with the electric spark, right? The primeval atmosphere. Okay, and the last one is direct observational evidence. So this is the fact that evolution still happens today. And in fact, there's a famous experiment run by Richard Lenski at Michigan State. He's had bacteria growing isolated from each other in these little flasks for years now. And in fact, since the 80s. And they've gone through about 75,000 generations isolated from each other. And what he's found is that in that time, you might notice there's one flask here, A3, is a little cloudier than the other ones. Well, something weird happened, right? So they put food in there, and then every day they would take out some bacteria and transfer them over to the next flask. And so every day they would get a sample, be moved on, and that sample would get a new food supply. And they would grow, and then the next day, some of them would get moved to the next flask, and so on and so forth, every single day since the 80s. But there's some other stuff in there too. There's a preservative called citric acid, and E. coli can't break down citric acid. But one day they saw this flask was cloudy, and it meant there was extra bacteria in there, more than would be expected. And what they found was that those E. coli had evolved the ability to digest citric acid, which is really unusual. So there have been lots of mutations along the way. In fact, they have frozen fossil records of each of these. So every 10,000 generations or so, they would freeze a sample. They can go back and look at the DNA, and they can see when different mutations took place. And this particular one, A-3, actually it took 2 mutations in order for them to get that ability. So far, they're the only flask that has evolved that, although all of them have gotten better at utilizing glucose, which is the normal food supply that they give them. So you guys are also, I'm sure, familiar with antibiotic resistance and the idea that bacteria have evolved the ability to outdo all of our antibiotics pretty much. And of course, viral evolution. I mean, how did the coronavirus get to be so bad? Well, it wasn't an infectious virus for humans. It probably came from bats. and then it mutated to be able to transmit from bats to humans. And then, I mean, this has happened before, but it made different mutations in other viruses that weren't quite as bad, that weren't able to spread as easily. But this one is able to spread really well from person to person, and so it just took off, and now we've got this worldwide pandemic going on. So, okay, and I do want to point out that despite what some people say, oh, where's the missing link? There are actually more fossils showing the evolution of humans than there are of T-Rex. So we've got all kinds of stuff, and along the way they found some different species. Like they know that some of us actually still have Neanderthal DNA in us. There's another one that's not on here called the Denisovans that were found in Asia. And the evidence really is overwhelming. So if you get a chance, go out there, take a look at some of the videos. I know I asked you guys to watch the Your Inner Fish series. It's really interesting. If you get a chance, watch that. So reflective practice, I want you guys to answer these questions. So how could these types of evidence be used to trace the evolutionary history of Homo sapiens? So the 5 different types of evidence: geographical, geological, physical, biochemical, and direct observation. So hope you're doing well. Thanks.